Okay, so I much appreciate the comments that Ford opened this event with concerning the integration of science and philosophy. I'll be trying to do a little bit of that. So evolution produces, of course, new kinds of plants and animals, and it also produces new kinds of things. So the multicellular organism, the eusocial insect colony, the eukaryotic cell, and so on. So some of the major transitions in evolution concern the appearance of these new sorts of things. And some of what I'm going to be saying is guided by the classic book that Maynard Smith and Zath Murray wrote about the evolutionary transitions. Along with those sorts of biological objects, other things as well arise. For example, with the multicellular organism, the organ arises as a part. And I think the gene itself as an evolutionary unit is brought into being by sex and other kinds of recombination of genetic material. So many different kinds of biological objects are made by evolution, and these have grey areas and movement between them, and I'm interested in general principles about those objects and the changes between them. Now, the way I'm going to approach this is by approaching it through the theory of evolution by natural selection. So I accept a sort of standard three-part Lewontin-style summary of what evolution by natural selection involves: variation, heredity, and differences in reproductive output. Any population of entities that fits those criteria, I take it, can evolve by natural selection. Now, a summary like that takes for granted various things, but most importantly, the notion of reproduction. Now, people often think about reproduction and fitness and related concepts in genetic terms exclusively, and some of the literature on evolutionary transitions has been guided by that idea. I want to generalise to all of the things that can generate parent-offspring lineages, especially lineages that feature parent-offspring similarity or some kind of heredity. Anything that can reproduce in a sense relevant to a Lewontin-style summary. So those are the things I'll refer to as Darwinian individuals, things that can reproduce in that sense. And then I think when we ask what sorts of things those are, we find three fundamentally different kinds of reproduction. I call these simple, collective, and scaffolded reproduction. All ways in which objects can form parent-offspring lineages with heredity and hence evolve, although with different mechanisms operating in each case and different kinds of borderline cases. So firstly, starting in the middle, a simple reproducer. I understand this as an object that can give rise to more objects of the same kind, make more things like itself, through its own internal machinery for the most part, of course in conjunction with external sources of raw materials and energy, but mostly through its own internal machinery. And also it's a thing that is not itself made up of other things that pass the same test. So in a hierarchy of things that all count as reproducing, it's the lowest level entity that can reproduce by means of its own internal machinery. So the paradigm here is a bacterial cell. A collective reproducer is a thing that can reproduce, but is made of things that can reproduce, which might be either simple reproducers in that first sense or smaller collective reproducers. So the paradigm here is an organism like ourselves. Thirdly, a scaffolded reproducer, in my sense, these are entities that reproduce or get reproduced, but this occurs by means of elaborate machinery of some kind which is external to them. So they can't reproduce under their own steam, putting it metaphorically. The paradigms here are things like viruses and also individual genes. So a simple reproducer, in my sense, can contain scaffolded reproducers, but it can't contain other simple reproducers, or it wouldn't be a simple reproducer. It wouldn't be the lowest level entity in the hierarchy that could do that thing. Now, these roles are somewhat idealised, and there are many intermediate cases, lots of grey areas. And I think that the evolutionary transitions involved, in many cases, moves between to and fro these different kinds of status. So in our case, for example, we are collective reproducers. Human beings fall into parent-offspring lineages. Our genes are scaffolded reproducers, and our cells Well, our cells are a special case for reasons that are central to this conference. Eukaryotic cells are some of the way to being simple reproducers, but they're also still, in other ways, collectives. So these are all different ways a thing can be a Darwinian individual, an object with a certain kind of importance to evolutionary theory. And these obviously include many things that are not organisms, genes especially. I think that quite a few discussions of individuality in biology run into trouble through not distinguishing sufficiently clearly the roles of organisms and the roles of these things, Darwinian individuals. Austin Booth has made this argument in more detail. For me, organisms are metabolic units, not evolutionary units. I'll come back to this later. Now, another kind of thing should be noted. As well as those sorts of objects, there are entities that recur without reproducing. So a simple example is a human heart or an enzyme within a body. This is a recurring object. They come and go repeatedly, but they are not brought into but hearts are not brought into being by earlier hearts in the way that cells are brought into being by earlier cells. So the hearts of your two parents do not have any kind of responsibility for your heart that your parents' other organs, their arms and legs, for example, had. Now, some of your parents' genes may have had a special relationship to genes in you that in turn have a special relationship to your heart, but genes are not hearts. And your parents' genes are linked in parent-offspring lineages to your genes, and they then, along with other things, through a developmental sequence, give rise to a new heart. So we've got various kinds of reproducing things and a category of recurring things which do not reproduce, where hearts and enzymes are paradigms. Now, the category of collective reproduction is an important one in evolutionary transition. So often the question is, when do you have reproduction at a new level as opposed to just reproduction of some smaller things, parts, in a way that has a certain kind of organisation? And this is where we find many puzzle cases in the literature. For example, debates about social insects, other kinds of social groups, ramets and genets in botany, and so on. Now, the right view, I think, is that there is a gradient here. With respect to this sort of Darwinian individual, collective objects that are evolutionary units in their own right, there's very much a distinction of more versus less. Now, I said gradient, but this is not really a matter of a single dimension. So in my book of a couple of years ago, Darwinian Populations, I gave an analysis of collective reproduction based around especially one set of problem cases. The paradigms for the analysis were the evolution of multicellular organisms from colonial forms and also the evolution of eusocial insect colonies compared to simpler insect colonies. The way I did this was distinguishing three parameters that define a space with respect to collective reproduction. Now, Mark talked about some of these in the previous talk. So the three parameters I used are the existence of a bottleneck between generations. So as Mark said, I think that bottlenecks or dramatic reductions in mass and structure have a special role in demarcating the beginning and end of a collective reproducing object, both because they restart a developmental sequence and because they generate close relatedness within collectives, especially within cells, especially between cells. Now, a bottleneck need not be a matter of a reduction to a single-celled stage, as in Bonner's and Dawkins' original discussions of the importance of bottlenecks. I think of this as very much a matter of degree. There are various different degrees of narrowing. A second parameter is germ-soma differentiation, which on the chart there is symbolised with G. I take it that the role of germ-soma differentiation is well known here. It prevents within collective conflict from having long-term consequences. Thirdly, overall integration of a collective I take to be important. This is inevitably a sort of a somewhat vaguer category, and I include it in the analysis in part to handle problem cases, which are not handled well by the other two parameters. So the idea between the analysis I gave earlier is that clear cases of collective Darwinian individuals, collectives that can reproduce in an evolutionarily relevant way, have high scores with respect to all three of those things, B, G, and I. And there are various interesting intermediate cases. I'll leave the slide up there, but I won't go through the various cases. I can discuss them if necessary. Now, as a multicellular collective like something like us becomes more integrated as a Darwinian individual, the cells within us do remain reproducing objects. They still reproduce because cell division is a kind of reproduction. And any collection of Darwinian individuals can evolve to some extent, but the ways that they do it will differ. And the cells within a body like our own bodies are partly de-Darwinised, especially by having the variation within the local population so much curtailed by the existence of the bottleneck between generations. It's also the case that reproductive differences between individual cells within a body have limited effects because of germ-soma differentiation, and the fate of a cell within a developing body is determined less and less by intrinsic heritable differences between cells as an organism becomes more complex, and more by control systems within the organism. So these are all forms of partial, not complete, but partial de-Darwinisation of these things, cells that were once clearer cases of Darwinian individuals. Now I'll start to head towards the topics of this conference by making a couple of general remarks about symbioses. So I distinguished organisms from Darwinian individuals a little while back and said that I think of organisms as metabolic units rather than evolutionary units. Now in some symbiotic associations, the partnership comprises something like an organism, I think, without being a collective Darwinian individual. Now, these are cases of tight associations between a host and a horizontally acquired symbiont. So a good example, a famous one, is the squid-vibrio symbiosis, the Hawaiian bobtail squid, which contains bacteria, horizontally or environmentally acquired, not vertically transmitted, which light the animal up. Another example might be a cow plus its gut bacteria, perhaps even ourselves. Lynn Margulis's term holobiont is often used or can be used for these things, for an animal or plant plus its microbial symbionts. And I think that if the symbionts have a free-living state and the holobiont is brought about by a combination of reproduction by the animal, say, and colonisation by the microbes, then the holobiont can be a recurring object that does not reproduce in its own right at all. It's not a Darwinian individual, even though it looks like one. These complexes reliably recur through the actions of several distinct Darwinian individuals that co-evolve but don't fuse into a single reproducing object. And this is how I think of the biofilms that Mark was talking about in the previous talk. So while there are examples of Darwinian individuals that are not organisms, there are also probably, or at least partial cases, of organisms that are not Darwinian individuals. Now, there will of course be grey areas and borderline cases. Associations of this kind have different degrees of tightness. Many symbioses are looser. This was one of the valuable things about the talk just prior to Mark's. So antacacia associations are a good example of a reliably occurring but somewhat looser symbiotic association. Where transmission is vertical, there is then fusion of the parts into a single reproducing object. So the aphid-Buchnera symbioses, which have been discussed several times in the last few days, would be an example of that. Another interesting case is a protist which mixes vertical and horizontal transmission, or rather acquisition, of an algal symbiont. So on cell division of this protist, one daughter cell gets the symbiont from its mother, and the other daughter cell must acquire a new one directly from the environment. Okay, having discussed symbioses a bit in general, I'll now start talking about endosymbiosis and organelles. Now, the way I'll do this, and the way I'll link it to the earlier discussion, is to make use of a distinction made by David Queller in 1997 in a book review, a review of the Maynard Smith and Zathmari book. So talking about transitions in general, Queller distinguished two kinds of transitions, which he called egalitarian and fraternal transitions. I don't think the terminology was that well chosen. I mean, Queller was being the book review was written in a somewhat light manner, and he was being cute with this terminology. It refers back to the French Revolution. The basic idea here is some transitions involve the association of like partners, and some involve the association of unlike partners, where the fraternal transitions are the ones where you have similar entities to begin with, such as the cells within a multicellular organism, who come together and form a larger unit, the advantage gained is initially just size and later division of labour. And the problem is well, according to Queller, the problem was that there often isn't much of an initial advantage, he thought. The egalitarian transitions, as he thought them, as ones where you have very different entities to begin with who come together. And the advantage is that there'll be a division of labour that's useful from the start. The problem then is he thought, was overcoming conflict. More exactly, I think, in both cases there is there's an issue of conflict that has to be overcome, where the difference is that in the fraternal transitions, such as eusocial insects, multicellular organisms, animals like ourselves, it's a problem of social evolution, essentially, to get the cells to cooperate. In the case of an egalitarian transition, such as, the ones that we're talking about at this conference, the coming together of very unlike things, such as two or more prokaryotes to give rise to a eukaryotic cell, then it's a kind of ecological conflict rather than evolutionary conflict that is the relevant one, because they're coming from different populations. Anyway, so let's look at the cases discussed in this conference through the lens of the Queller transition and connect it back to my earlier framework. So the framework I described earlier from my book was very much shaped by the fraternal cases, and there's a question about how it might apply to the egalitarian cases, especially the evolution of the eukaryotic cell. So let's assume, perhaps as an idealisation, that we have a simple kind of engulfing scenario where one prokaryote engulfs another, eventually giving rise to something like a mitochondrion, perhaps as a one-time event, perhaps not as a one-time event. Now, in cases like that, there are a series of shifts between the categories that I described above, which are different from the ones you see in fraternal transitions. We start with two simple reproducers, two prokaryotic cells. It's the paradigm for me of a simple reproducer. They then form a certain kind of collective, initially a very loose one, because all you have is a kind of engulfing, and some degree of integration may then come along. It may also then be the case, including as seen in mitochondria in animals like us, that what was once a simple reproducer, a microbe, moves some of the way to being a scaffolded reproducer, because it loses the ability to reproduce by means of its own internal machinery. If it gets to that point, then the cell that contains both partners, is then a new simple reproducer, not a collective. It's now the lowest level entity in the hierarchy that can reproduce under its own steam without having parts that can do the same thing. It's also possible in principle for organelles to become a recurring structure that doesn't reproduce at all. But that I take to be something not seen in mitochondria, and I don't know if it's seen anywhere. Now let's talk a bit more about each of these steps. So firstly, in talking about the early stages, there's a crucial difference between the fraternal and egalitarian cases. In the fraternal cases, multicellular organism, eusocial insects, we have a collection of similar parts, and by organising their activities, a collective gets a new status. And in all those cases, it's essential that one part can stand in for the others in reproduction, by means of especially things like germ-soma differentiation. In the case of endosymbionts, there's no way for one partner to stand in for another in reproduction. When there's a population of endosymbionts within a cell, then some of the same factors that are relevant in the fraternal cases are relevant again. So here I can refer back to Steve Perlman's talk about mitochondrial inheritance. And also, even with uniparental mitochondrial inheritance, there can be conflict issues to overcome in these cases, because at least in animals, high mutation rates in mitochondria make mosaicism an issue, and hence evolution in the population of mitochondria within an individual. Some of the mechanisms that deal with this in organisms like us do seem to have a real analogy to the mechanisms seen in the fraternal transitions, especially the existence of various kinds of partial bottlenecks. I won't go on in more detail about that now. Because that doesn't bear on the basic mitochondrian host relationship, where neither party initially can reproduce on behalf of the other. And so of the three factors that I mentioned earlier in my analysis of collective reproduction, only integration is relevant to this relationship. A strong form of integration of that kind is, of course, transfer of genes from the endosymbiont to the nucleus. So let's now start thinking about the stages that occur later in the sequence when you start to have a loss of endosymbiont genes to the nucleus of the host, or even if the host doesn't have a nucleus, just to the host, where those genes are important in making gene products that go into making a new symbiont, such as a mitochondrion or protomitochondrion. The transition that is then beginning is a transition towards what I call scaffolded reproduction. So you reach a situation where an existing mitochondrion gives rise to a new one, which is an offspring mitochondrion. It was made by fission with a determinate mother. There was a fission event, but the making of the new mitochondrion relies on physically external machinery in a way that was not the case in the earlier free-living state and the earlier stages after absorption. The evolution of new nuclear genes that make mitochondrial proteins even without transfer also achieves this. So in my way of looking at things, a scaffolded reproducer, again, is the paradigms are things like genes and viruses. A mitochondrion is making its way towards being a distinct form of scaffolded reproducer. It has a unique parent, a unique parent instance, but its reproduction is dependent on external machinery. Unlike the case in other scaffolded reproducers, though, the handoff of nuclear genes, the handoff to the nucleus, or the handoff to the host of endosymbiont genes in this case, reduces the evolutionary capacity of the mitochondria. In particular, it will reduce heritability from one mitochondrion to another. The fission events themselves won't be associated with significant heritability in the evolutionarily relevant sense. So earlier I said that in transitions, for example, in the transition to a multicellular organism or to a eusocial insect colony, the parts are de-Darwinized to various degrees. Their evolutionary capacities are often curtailed, not entirely but partly, by the events that give rise to a higher level unit. And here, the dependence of mitochondria on host genes does this in a different kind of way from the ways discussed earlier. Now let's consider what happens as the mitochondrion loses more and more genes to the nucleus. So more and more of the rebuilding of the new mitochondrion or other endosymbiont is being handed off to other entities. And this might not just be to the host nucleus. So in John Archibald's talk, we heard about some interesting cases with secondary endosymbiosis, and in particular in the cryptomonad that he works on. 4 genomes are present, and the chloroplast needs proteins. To make a new chloroplast, or make a functional one, it needs proteins synthesized by 3 different genomes, its own, the nucleomorphs, and the host. Focusing so it needn't just be handoff to a single external object, but perhaps to many. Focusing, though, just on the mitochondrial case, where there's one symbiotic event, but lots of gene loss. As it goes along, I'm suggesting that it's less and less the case that a new mitochondrion produced has a parent mitochondrion. There's less and less determinate apparent offspring lineage between those objects. Instead, to a greater extent, as the process goes along, a new mitochondrion is made by the whole cell. It's one of the many things made by the whole cell. In the limit, it could become another case of the other category of object I discussed earlier. It could become a recurring object, which doesn't reproduce at all, like a heart or an enzyme. Things that come and go but don't have parent-offspring relationships between instances or tokens. Now, that's only true in the limit, though. It's hard for a scaffolded reproducer of this kind to lose reproducer status completely, because this would require not only that all the genes be lost to the nucleus, as does happen in some cases with mitochondria, but also another basis for parent-offspring lineage identification exists, which is the membranes themselves. So in mitochondria, Membranes also provide a basis for at least the bare presence of a parent-offspring relationship between one mitochondrion and another. Each mitochondrion has a unique mother with respect to its membranes, even if it got a lot of its proteins from somewhere else. So it's hard for an endosymbiont, at least of the sort we're talking about here, I take it, to tread the complete path from simple reproducer to scaffolded reproducer to a recurring object which doesn't reproduce at all. I'm curious to hear about whether people have in mind cases that perhaps get close. But regardless of whether the difficulty in reaching this sort of possible final state here, as the process goes along, more and more of the important properties of a new item, the mitochondrion, will not be due to the parent mitochondrion. The causal role of the parent gets reduced more and more as the mitochondrion contains more and more gene products that derive from the host nucleus. And in evolutionary terms, you can think of that as a steady loss in heritability with respect to that parent-offspring relationship between one endosymbiont instance and another. Bill Martin in his talk said, what's the difference between an endosymbiont and an organelle? Don't care, he said. I certainly agree with respect to the words. We shouldn't worry too much about where exactly the border might be put. But it's also true that a heart has a different evolutionary role from a cell and different again from a gene, and there can be transitions between those different kinds of objects. The paths are important, even though the labels we might apply to various pieces of the path are less important. And a eukaryotic cell, I take it, is at a very special place with respect to these roles. And that's due to the facts about which objects retain parent-offspring relationships and what kinds. Okay, I'm just about done, so am I pretty much at the end of my time? Or another 1 minute, good. So I'll just summarise, I'll summarise again. So let's just do the sequence again while noting all the objects that have parent-offspring relationships and all the objects that don't and what sort they have. So we start with two free-living Darwinian individuals, simple reproducers, prokaryotic cells. They contain chromosomes, which are scaffolded reproducers in my sense. One engulfs the other. Initially then, we still have two simple reproducers and a very low-grade collective, something which is unified just by spatiotemporal location, linked only by containment. But one possibility is then that you get a kind of integration to some degree of those, perhaps synchronization of the cell cycles, perhaps signaling of various kinds, and the combination becomes a collective reproducer in the richer sense. That's one of my targets here. As integration continues, the endosymbiont moves away from simple reproducer status to scaffolded reproducer status, as it comes to depend more and more on external machinery. If we decide that the endosymbiont is entirely a scaffolded reproducer, then the combination, the eukaryotic cell, is then a simple reproducer. It's then the lowest level object in the hierarchy that can reproduce under its own steam without containing other objects of the same sort. And then in the limit, it could become a recurring object which doesn't reproduce once the parent-offspring lineages are lost. But again, that would require not just loss of genes, but loss of all the other determinants of parent-offspring relationships, such as parent-offspring length with respect to membranes. So I guess it would be possible for an endosymbiont to become an organelle in a sense that made it entirely akin to an organ like a heart, something which is rebuilt, something which recurs, but does not reproduce. It would then travel a path from simple reproducer to part of a collective to scaffolded reproducer to a thing that doesn't reproduce at all. Now, in actual cases, I take it, at least with mitochondria, they tend to stay somewhere else along this line. And then, at least in mitochondria, you then get further effects and dynamics that occur if the eukaryotic cells, which are now part simple reproducers, part collective, then give rise to multicellular organisms in a further evolutionary transition. But that, of course, would be a topic for another talk.